Hello, welcome back, everyone. Hope you had a refreshing break, and we are thrilled to have you to re-join us for the continuation of the seventh Brazilian Student Council Symposium on Neglected Disease. We hope your lunch was delightful and you're re-energized for the exciting season ahead. So, our first short talk session will be presented by Lissur Azevedo Rosini, graduated in biological science from the Federal University of Minas Gerais, master and PhD in bioinformatics from from the same university, with a sandwich period at Queen Mary University in London. Second. During her undergraduate students, she completed her undergraduate research at the Molecular Pathology Laboratory at, at the same university, in the School of Dentistry, where she worked with molecular biology applied to the study of head and neck tumors. During her master's degree, she investigated the molecular mechanism involved in the biology of the mammary gland, as well as its evolutionary origin. And during her PhD, she investigated the gene expression profiles in human tissues with focus on development of metrics for distinguished genes relevant to tissue biology. Currently, she works as a bioinformatician at the Laboratory of Molecular Parasitology at the Faculty of Medicine of Ribeirão Preto at the University of São Paulo, where she has dedicated herself mainly to the study of gene expression in Leishmania brasiliensis. Today, she will present us her project named Evaluation of the Effect of PRMT5 Knockout of Leishmania brasiliensis transcriptome. So, welcome, Sue, and the stage is yours.
Okay, thank you, Matheus. So, I'm Lissur Orsini, and I'm going to present the work evaluation of the evaluation of the effects of PRMT5 knockout on Leishmania brasiliensis transcriptome, led by Gustavo Campanharo and supervised by Angela Kaisel Cruz. So, who is Leishmania? Leishmania is the protozoan parasite responsible for leishmaniasis, a group of disease characterized by a high degree of diversity presentations, subdivided mainly in tegumentary, both cutaneous and mucocutaneous, and visceral. It's endemic in 98 countries, and over 1 billion people are at risk of infection and is considered a neglected disease. In special, Leishmania brasiliensis is the main causative agent of tegumentary leishmaniasis in South America, and it can cause the mucocutaneous form, this one, which can lead to the destruction of mouth, nose, and throat mucosas. And who is PRMT5? PRMT5 is one of the protein arginine methyltransferases. That means it methylates arginine residues, and a common target of PRMTs is RNA binding proteins, which in turn conduct to post-transcriptional gene expression regulation. From the 5 PRMT PRMTs known in L. brasiliensis, it was already demonstrated that the absence of PRMT5 compromised the in vitro infection of macrophages. Thus, our objective was to perform RNA-seq analysis for PRMT5 knockout. For that, we extracted RNA, total RNA, from the infective flagellar form of Leishmania brasiliensis, this one, for both the knockout and the wild type, but also for the add-back cell line. Add-back is when you remove the gene, PRMT5 in this case, and then you reintroduce the gene to check if the phenotypic changes observed are kept. And then we perform RNA-seq of parent and strain-specific RNA-seq with a coverage of about 70 million reads per library. We conducted a differential analysis using our bioconductor dissect2 package, and we considered a gene as differentially expressed when p-value adjusted was less than 0.05. To try to gain biological insight, we also generated an interactions network in StringDB, using as input both PRMT5 and their respective differentially expressed genes, and also a set of proteins known to physically interact with PRMT5 in Leishmania brasiliensis. We found that 99% of Leishmania genes were expressed, and of which 27 were differentially expressed. When applying a fold change of more than 1.5, we found 3% of the expressed genes as differentially expressed. In general, the number of upregulated genes was greater than the number of downregulated genes, as we can see here, but also in the Vulcan plots shown here in red. And also the upregulated genes exhibited lower values of p-value adjusted and larger values of fold change. That means they were more significant. When we look at the add-back versus wild-type comparison, we found a small number of differentially expressed genes, specifically only 3 upregulated genes. This is an indicative that add-back restored wild-type. That means the phenotypic change could be restored in the add-back cell line. And we can also see here in the Vulcan plot only 3 upregulated genes. And this means the wild type is similar to the add-back cell line. We also found a chromosome bias in our data. So from these 80 differentially expressed genes shared between comparison of knockout versus wild type and knockout versus add-back, 73 or 83% were from chromosome 27, and they were distributed throughout the chromosome, as you can see here, the entire chromosome 27, with these genes highlighted in pink. And we did not find functional relationship between these genes. So why we have this bias in our data? Our first hypothesis was chromosome 27 could have an extra copy in the knockout, because this is common in Leishmania. But we already checked that and know the knockout is deployed. And so we are still investigating this bias. Finally, we generated an interactions network of 157 protein and 328 interactions, and we could identify at least 3 functional clusters, nucleolus and ribosome biogenesis in eukaryotes with 14 genes, proteasome and regulation of cell cycle with 11 genes, and carbohydrate, amino acid, and fat acid metabolism with 17 genes. Uh, we, we are conducting some phenotypic assays. Basically, we evaluated the ability to transform in the intercellular form of Leishmania brasiliensis for these 6 downregulated genes shared between these 2 comparisons. And we got a positive result for 2 of them. As perspectives, we are still investigating the chromosome 27 bias, and suggestions are welcome. And we are still conducting some phenotypic assays. I would like to thank all my colleagues from Laboratory of Molecular Parasitology, and special Dr. Gustavo Campanharo, who is the main author of this work, and Professor Angela Cruz, which is our supervisor. I would like to thank the funding agency, FAPESP. I would like to thank the organizers for inviting me, and I would like to thank all of you for listening. Thank you.
So thank you, Lisu, for the amazing presentation. And now let's open the floor to the audience. So if you have any question, please share them in the chat, and we'll navigate through them during the questions and answers right now. Just wait a moment. Well, first I can do my own question about the question that someone asked previously in the last presentation about the biggest struggle during the research. Which one was for you during this project?
I think the information available, because it's a challenge to work with Leishmania. I didn't work with Leishmania before, and all the databases are not complete, and the tools, many times you need to adapt the tools for working. Like a half of the proteome is hypothetical, so it's a challenge to work with Leishmania in general. And I think in this work particularly is difficult to work with the infective form and the intercellular form, because you have a low amount of transcripts.
Thank you. We have our second question projected here. Elvira asked, what do you think happened 3 large groups in the indicated network, the strings?
We are evaluating these groups, these clusters, and some of them we think is related with this transition from the flagellar form to the intercellular form. Like you have a change in the complete metabolism, so the proteasome is meaningful for us. But I don't know, do you think should have more than 3?
Yeah. If she answer, I will show you there.
Okay.
So just because of the time, let's go to the next question. So Alexis said, great presentation. Have you evaluated the efficiency of the PRMT5 knockout and delivery for producing the groups in the analysis?
Uh, Not yet. Do you mean the networks analysis, right? This is a recent um, analysis, so we did we didn't have time to evaluate the clusters there, but we're investigating some of the differentially expressed genes, and some of them are presented in the network. So this is currently ongoing. I don't know if it's answer your question.
So I think we need to stop right now because of the time, but thank you once again, Lissur, for your amazing presentation.
Thank you.
And now we're going to our next, just one second. Uh, here. So we go for our next presentation, and we have Raíssa Medina Santos. She has a degree in biomedicine from Minas Gerais Education and Culture Foundation, the FUMEC, with qualifications in molecular biology and clinical analysis. Master in bioinformatics from the Federal University of Minas Gerais, and PhD in genetics in genomics, no, genetics and bioinformatics from the same university, with a 12-month samplage period at the Centre National de la Recherche Scientifique in Paris. Nowadays, she's a postdoctoral researcher at the Conservatoire National des Arts et Métiers, sorry for my French, in Paris, and she has experience in the fields of biochemistry, immunology, and bioinformatics, focusing on transcriptomic, venoms, and toxins of spiders, scorpions, and snakes. And today she will present her project named Gene Expressions Profiling in Chikungunya Infected Patients, Reveal Potential Mechanism of Chronic Symptoms. Welcome, Raissa. The stage is yours.
Thank you. Good afternoon. My name is Raissa Medina. I'm a postdoc at Laboratoire de Génomique, Bioinformatique et Chimie Moléculaire, le Conservatoire National des Arts et Métiers at Paris in France. And today I will present our latest results entitled Comparison of Transcriptome Profiles between Patients with Chronic Symptoms post-chikungunya and no chronic controls. This work is a collaboration with the Centre Hospitalier Universitaire from the La Réunion Island, l'École d'ingénieurs généralistes en informatique et technologie du numérique, and the Hôpital Henri Mondeur, both from France. So a little introduction about our work here. Our subject is with the chikungunya virus, which is a tritogenic alpha virus, and his vector is the mosquito Aedes. So in all the world, we have a large epidemics, especially in 2005 and 2006, where we have a large epidemics in La Réunion Island, where more or less 40% of the population was infected. There's a lot. There's other epidemics are also known, such as in 2007 in Italy, and especially in 2013 and 14 in the Americas, especially in Brazil. So after the patient are infected with chikungunya virus, he presents two phases of symptoms, named acute and chronic. And in the acute phase, so is the first phase of the infection, in the first to the 21st day of the infection, more or less 80%, according to the population of patients, present a few symptoms such as fever, joint pain, rashes, vomiting, and headache. And if the patient continues to present symptoms after the third month of symptoms, he will consider it a chronic patient, where more or less 50% of the patients will present some symptoms. The more no symptom in the chronic phase is the post-chikungunya chronic inflammatory rheumatism, which is very similar to rheumatoid arthritis, which is an autoimmune disease. But he also can present chronic fatigue, light cerebral disorders, headache, mood disorder, and all that can deteriorate the health-related quality of life of the patients. So since chikungunya infection is a major public health issue and several questions around the chronicity are made, our objective is to understand the molecular etiology of the chronicity after the infection. And as a specific goal, we want to compare the transcriptome profiles of PBMC between chronic and no chronic patients, focusing on the identification of the molecular signatures from chikungunya symptoms persistence. And it's important to say that this is the first transcriptomic study ever long-term symptoms following chikungunya infection of 12 to 15 years hindsight. So our cohort is from patients of the La Réunion Island Hospital. We have a big cohort of 600 patients, but for this part of the work with the transcriptomic, we will use 136 chronic patients versus 88 no chronic patients. Between them, we are male and female patients. So in our methodology, the first step is the patients who was infected in 2005 and 2006 come back to the hospital in 2018, 2020 for a blood collection. And after that, we made a PBMC isolation and RNA extraction and sequencing using Illumina HiSeq. So the third part was a bioinformatic analysis where we start with FastQC and MutQC for the quality of the samples, the star alignment for the alignment, the feature counts for the counting, the SEC2 for the analysis of the genes differentially expressed, and string and Metascape for the pathways. So we will have a little look in our latest results. So here I present a volcano plot where we can see the differentially expressed genes between chronic versus no chronic patients. We have more or less 2,000 genes. And if we look a little more in the top 20 genes, so the more relevant genes in this comparison, we can see we have relevant terms in viral infection and immune response. After that, we also have another genes who are not theoretically very focuses in a viral infection, such as olfactory signaling pathway and epidemical growth factor, but this also can be interesting for other types of research in this team. So after have our list of genes, we follow the next step, who is the pathway analysis. For that, we use Metascape. I use the significant list of genes that I showed before, and we check the top level enrichment terms. So here we can see that there is a lot of terms connected to the RNA. So we have metabolism of RNA, the no coding RNA metabolism process, but we also have another pathways who are very interesting, such as cellular response to stress, DNA damage response, regulation of cell cycle process. With the same group of genes, we can also check using Metascape which of these genes are connected with some disease. So we use the GisGenet inside of Metascape. And it's very interesting to see in these pathways, we can see a lot of pathways connected to the rheumatoid arthritis. And this is very interesting because the majority of our chronic patients have present symptoms that are very similar to rheumatoid arthritis. And this can show there is really a gene level of connections between the symptoms of the chronic patients and the real rheumatoid arthritis. In the last part, we also check using string, who use all the ranked gene list to see also if we find another top level enrichment partners that can be interesting. And for that, we also find pathways connected to RNA, to the connection with the viral protein interaction. So we have viral protein interaction with cytokine and cytokine receptor. But a pathway that we thought very interesting to find is morphine addiction. So why we thought that this is a pathway interesting to check? It's because in the treatment of arthralgia in the acute phase of chikungunya, when the patient presents severe pain, he is treated with dipyrone or paracetamol combined with an opioid, such as tramadol or codeine. And opioids already know for influence the viral replication and the immune function, and this can impact the disease progression. Maybe positively or negatively. So maybe we think that we can have a possibly connection between the use of opioid in the acute phase and maybe presence of these in the cases of patients who will be chronic. But this is very beginning of our results in this part, and you need to be very, very investigate in our cohort. So is what we're doing now. We are checking if which patients was treated with opioid, which type, when, how much, and this is just the beginning of this research. So in conclusion, this is the first transcriptomic study on chronic symptoms following chikungunya virus infections to 12 to 15 years hindsight. The analysis of the genes differentially expressed show a strong role for immune genes on chronicity, and this can start to give us a response to the question if there is antigen persistence or a latent viral reservoir. We also check that in the disease pathway analysis, we retrieve the implication of the genes involved in arthritis. That is very interesting and corroborate with our research or chronics with the patients with signaling of arthritis. In the biologic pathway analysis, we check that we have a pathway with RNA transport pathway that mirror the fact that chikungunya it is a RNA virus. And the viral protein interaction and cytokines pathways confirm the role of the immunity. Both of these pathways also help us to try to understand the latent viral reservoir. And in the end, the morphine addiction pathways raise an intriguing question regarding the use of opioids in the acute chikungunya treatment and its potential impact on the disease progression. But as I say, it needs to be more investigated. So in our perspective is to have more additional analysis on our transcriptomic data, so the weighted gene co-expression network analysis, also the cell composition deconvolution, and we also want to evaluate the impact of the sex patients in your data. Besides that, we are also working with the Genome Association study of this data and correlated with the transcriptomic data. So I would like to thank everybody for their attention. I would like to thank our laboratory, GBCM, and the Centre Hospitalier Universitaire de la Réunion. Our financial agency, it's the Agence Nationale de la Recherche, and of course, the student group that give me the opportunity to talk about my work. So thank you very much.
Thank you, Raíssa, for the amazing presentation. Now let's open the floor to our audience. So if you have any question for Raíssa, please let in the chat below and we will project your question. And first I have a personal question again. I have a colleague in Brazil that works with chikungunya, with chronic chikungunya, and the most difficult part for him is about the samples, because it's really difficult to find someone with chikungunya who had the right diagnostic. And so my question is, how does it work in France for the mapping of patients with chronic chikungunya?
Yes, so in this case, we have the Dr. Patrick Girardin, who lives in La Réunion Island, and he worked since the start of the infection in 2005 with the patients. So he have the real contact of the patients, and he passed many years working with this data. So he has all the control from the beginning, from the first day that the patient start to have the first contact of the disease until now, if he's still alive, but because maybe some patients can also be be dead after so many years, and he had all the control of all of that. And we have very, very lucky, because it's really very difficult to control this type of information. And we are very grateful, because it's really a great chance to have all this data available with him to work.
So we have one question in the chat. Mateus asks, very good presentation. Thank you. Do you investigate the presence of no-coding RNAs, mainly long no-coding RNAs? If not, do you plan to do that?
Yeah, it's a very good question. In the beginning, we don't think about it. But after we check that we have a pathway who talk about that, we start to think, yes, maybe something that we need to also focus. So yes, is it in your plans? But I didn't start with this part yet.
Thank you. And we have another question. Bibiana asks, do you expect difference between sex and age patients' outcome?
Yes, actually, it's very famous that we have difference between sex and transcriptomic data. So normally, yes, we can find the differences. Also with age of patients, the age of patients is very important normally in the transcriptomic data. Actually, it's not presented here, but I already made this type of analysis, and we find differences more expressed in females, and that corroborate maybe the importance of the females that have more chronic cases and more problems in the chronicity than men. But that's a lot of other results. I didn't have the time to show that here, but yes, sex and age are very important in the data, and should take in consideration.
Thank you. Maybe our last question, because of the limited time. Elvira asks, what part of the results do you believe will help in the treatment or quality of life of the patients?
So we have this, I talk really with not sure, but we have this morphine pathway that it's very interesting, and it's really, really difficult to say by sure. But with this morphine pathway, maybe it's not the best treatment for the patients. So it's very interesting. That's why we are so focusing and work more with this information, because if the morphine, it's really something that can change the progression of the disease, maybe all the treatments should be changed. But again, it's very, very beginning of something, so I can't say anything for sure in this moment.
Great. So thank you once again, Raissa, for the presentation. And now we'll follow for our last presentation. Thank you.
Thank you.
So now, finally, but not least, we have Tangitênio Lima de Souza, graduated in biological science from the Federal University of Espírito Santo, master and current PhD student in cellular and molecular biology at the Federal University of Paraná in the field of cellular and environmental toxicity. He has experience in reproductive toxicology, endocrine disruption, oxidative stress, morphology, and cell culture. And today he will present us his project named Understanding the Mechanism of Action of Hair-Grown Stimulating Compounds on Hair Follicular Dermal Papilla Cells as a Model for Alopecia. So welcome, Tom Jenny, and the stage is yours.
Hello, everyone. Good afternoon. My name is Tugistenio, and I'm a PhD candidate from Federal University of Paraná. And today I will present to you part of my work entitled Understanding the Mechanism of Action of Hair Growth Stimulating Compounds on Hair Follicle Dermal Papilla Cells as a Model for Alopecia, developed in collaboration with Grupo Boticário. First, we need to understand what alopecia is and its origins. This disease can basically be defined as a lost hair density or hair thinning, and this is a phenomenon caused by two main sources. It can be environmental, such as stress, or genetic. And there are basically four main types of alopecia. That could be aerata, cicatricial, diffuse, or androgenetic. So there are other types of alopecia, and the source of the disease can often be environmental and genetic mixed. So here I'm showing to you the general mechanism of how the disease occurs, and here we have the anagen 6 form, where we have the dermal papilla and the hair follicle in a health condition. However, due to the environmental and genetic factors that promotes alopecia, there's a regression of the hair follicle and its structures, which can be at viral levels such as catagen or telogen phase and other phases of anagen. And from then on, the hair no longer can develop it in this situation. And to evaluate the effects of alopecia and the compounds that we can use to treat this disease, several in vitro models have been developed, and we can use different types of cell lines as well, the combination between them to form 3D organoids, as shown here in the figure, where it is possible to observe a spherical bulb formed by basically keratinocytes, and other types of cells that can grow, forming and structure hair-like. And then in this way, we can mimic the extension hair and the treatment with some chemicals using these kind of models in vitro. So some synthetic compounds for the treatment of alopecia are available, but they have shown some side effects in patients. And based on this, the use of new therapeutic approaches using bioactive products to reduce the risk of hair loss has been emphasized by the literature. And the two chemicals more famous that are used today is minoxidil and finasteride. They are the best known drugs for treating this condition. And the mechanism of action of these compounds is related basically to the increase of blood and nutrient flow on the skin and the inhibition of some enzymes related to hormone metabolism. So in this sense, this work aims to use some transcriptomic databases from NCBI to identify biomarkers through functional analysis, enrichment analysis, to evaluate the potential treatment of some natural compounds in treat alopecia, using basically hair follicle dermal papilla cells as a study model to understand the mechanism of action of these compounds, and basically to prospect new biomarkers to analyze some efficacy of compounds. So for the methods, the experimental design is quite simple. We look for two data sets from NCBI GO, and these data sets, they were used and integrated. All the data were processed using our software and analyzed to identify the differently expressed genes between the control and the treated groups. The statistical analysis followed the standard protocol for transcriptomics studies by using microarray data sets and the Lima package. So revigor KEGG pathways and string servers were used for functional and enrichment analysis. And at the end, all the datasets were integrated and compared between them to evaluate the biomarkers according to the biological pathways of interest. So the experimental data and design from both datasets, they consist in 2 in vitro experiments where hair follicle dermal papilla cells were treated with 10 micromolar of TCKA, that is a natural compound, or they were treated with argan press cake extract, that is a natural extract from the argan, in a concentration of 50 and 8%, depending on the treatment. And the cells were exposed for 48 hours, and in both data sets, the RNA samples were prepared for gene expression profiling, use GenTube from Affymetrix. So here we have the results, and according to the results for the differential expression analysis in the volcano plots, we can observe that there are many up and down regulated genes. And when you compare especially when we compare between the control group and the treated one. So when we take a look at the Venn diagram, there are in total at least 3,000 genes in common that were differently expressed between these 2 data sets. And considering this result, the next analysis that I'm going to show you are going to be focused on these 3,000 genes. And here, the functional analysis of these 3,000 genes were analyzed by the Revigo web server, and the results obtained. In the results obtained, we can see that there are a lot of geoterms were analyzed by a multidimensional way and grouped in this graph, in this plot, and each bubble represents a biological process. And they were grouped into structural cellular chains, cell cycle and proliferation, macromolecule metabolism, especially the lipid metabolism, which is essential for the formation of hair egg structure, and signaling pathway. These genes were also evaluated for their molecular function, being associated basically with transport, enzymatic, regulatory, and binding activities. So in addition for cell components, we can see that most of these genes were constitute of protein complex and receptors for endoplasmatic reticulum and mitochondrial matrix. And the pathways that were analyzed were focused on 3 main pathways. The first one is the extracellular matrix interaction, which is a pathway of great relevance to the development of hair. And here we have the KEGG output result, where all the red genes that are highlighted here is one of the differently expressed genes. But when we take a look at all these genes between the treatments and the different data sets, we can see that only these ones here were equally modulated between the 3 treatments, being up or down regulated. And among these ones, the integrin B8 and THBS1 are the genes that are most important in modulating hair growth, and they can be considered good candidates to be evaluated of the efficacy of some natural compounds on this kind of cells. The other one here demonstrating the results for the second pathway, that is the cellular senescence, which is extremely important for the vitality of the follicle and its maintenance in the hair growth. So in total, there were basically 8 genes that were equally regulated in the data sets and in the treatments. And among these, the insulin growth factor binding protein 3, that is this one here, is one of the most important that we could analyzed in this pathway, since this gene is also important in stimulating the hair follicle maintenance and also being considered a good biomarker candidate to analyze the efficacy and of some natural compounds on this type of model for alopecia. And the third and last pathway selected was the regulation of cell pluripotence, which is an extremely important pathway given that in the hair follicle, there are some stem cells that are actively responsible for the cell renewal. And in this sense, it's possible to observe that there was an increase in the WN2B and WNT7B genes that are genes important in the modulation of this kind of pathway, especially for development and differentiation of these cells. Also, we carried out a network analysis using the string server, and in this network interaction analysis, all the genes were highly connected, and the results demonstrate that they are connected to each other, and probably they could be acting in an integrated manner to regulate physiology and stimulate maybe hair growth and hair follicle in the dermal papilla cells. So the main conclusions of this work is the mechanism of action. Some results were no show, but the treatment with natural compounds, the TCKA and argan press cake extract, that are natural compounds, promoted changes in the genes that are important for stimulating hair growth and prevent hair loss, having a mechanism of action like the synthetic compounds that are commercially available, such as minoxidil and finasteride. And what is new about this work? It was possible to identify new biomarkers that were equally modulated between the different treatments, and these genes are important for maintenance of the hair growth, such as integrin BH, THBS1, insulin growth factor binding protein 3, and all these genes can be used to test the efficacy of some natural extracts or natural compounds that could be development, and it can bring new claims and innovation for hair products. And in this sense, the hair follicle dermal papilla cells as a model for alopecia proved to be promising for identifying the action potential of these compounds that can stimulate the hair growth. However, to analyze a more physiologically and realistic scenario, is necessary to develop a more robust 3D model for greater reliability of the results. And as next step, we are going to test some compounds and evaluate the gene expression of these biomarkers to try to validate the results. So that's it. Thank you for your attention.
So thank you very much, Eugênio, for the amazing presentation. And now let's open the floor to our audience. So if you have any questions, please let in the chat so we can project here and ask to Eugênio. So I have one personal question. Even though 3D cell cultures are fresh techniques and can mimic the tissue environment better than 2D culture, do you think they are enough for the transcriptomic analysis? Thinking about, for example, the epigenetic influence in the results, or do you have any further plans for validation with in vivo samples or something?
Actually, I don't know if the 3D models can be evaluated for epigenetic modifications in this context, but I know that I know that for analyze the hair growth and stimulating compounds, it can be used. There's a lot of models published, And the next step is we are going to try to reproduce them in our lab and create some modifications to evaluate the potential of some natural extracts and compounds.
Thank you very much. And now we have another question from Elvira. Which of the results were a positive or negative surprise considering current hair products?
Okay, let me think. Using natural compounds, one of the things that I could see, I didn't bring these results, but using natural compounds, a lot of beneficial pathways and the biological process were upregulated compared to minoxidil or finasteride, for example. So using this kind of results, we can bring some new claims to hair products and more beneficial, for example, the hair health. I don't know if this term is correct, but you can improve the hair condition compared to use synthetic compounds. So we have more benefits in this sense. The pluripotence one was surprising to me because I didn't see this type of results in the literature using chemical synthetic compounds.
Thank you. And another personal question. Do you have plans to integrate with another kinds of analysis? Could be with bioinformatics, structural biology, or I don't know, for the next steps in this program?
Okay. The structural biology, yes. I would like to run some, for example, docking or molecular dynamics analysis. To be honest, I do not consider myself a bioinformatician. This is one of my first works that I have developed in this field because I'm a toxicologist. So yes, the structural biology is like one of the analyses that I would like to integrate with all these kind of experiments.
Great, thank you very much. Our last question, because of our limited time. Bibiana asks, considering that alopecia is a condition of hair loss, is this model considered a control model for hair growth or maintenance of hair structure?
Okay, it can be used for both purposes, but the most used purpose that I have seen in the literature is the use for hair growth, because you can stimulate, you can use, for example, bioprinting and put some this kind of cells in a 3D model of skin, for example, and observe how they can respond to your compound. For hair maintenance structure, yes, it can be used too, depending on the gene or the pathway that you are going to analyze.
Okay, thank you. So that was our last question. So I want to thank you again, Doug, and one thought thank everyone from the short talk panel that we have today. Don't forget that we'll have another one tomorrow, and we'll have a prize, cash prize in the end of our symposium. So thank you once again, Doug. Bye-bye. So now we're gonna have a 3 minutes break before we get to our next presentation of the Professor Lucien. Okay, so if you want to have some water, go to the bathroom, now is the time, and we'll be right back. And yeah. So we are back to continue with our symposium, and now to finish our last day of our first day. Né? We have the Professor Luciana da Silva. She has a master and PhD in molecular biophysics from the São Paulo State University, UNESCO, and it is currently a postdoc and he has a postdoc in bioinformatics. Professor Luciani is a researcher at the Molecular Oncology Research Center and a professor at the postgraduation on oncology program of the Cancer Hospital of Barretos. She has experience with computational biology, with virtual screening of natural products, and system biology, analysis of microRNA, messenger RNA and microRNA expression, development of risk scores, and graphical interface for research tools and molecular diagnostics. So today she will present us the role of computational biology and virtual screening in combating neglected disease and in this presentation. So Professor Luciani, the stage is yours. Welcome. Presentation and
Okay, thank you. Thank you very much for the invitation and also for the kind introduction. It's a pleasure to be here today and know a bit more, learn a bit more about the RSG Brazil. I think it's my first time in this event. I can still have a lot of outstanding students here. So thank you very much again. I believe I'm less experienced than my fellow speakers in neglected diseases, but I have been working on different fields of computational biology over the years. So I would like to highlight some points about the importance of this kind of research and some topics about career in bioinformatics as well. So I'm really curious to hear your thoughts and questions and bring up some insights on career in bioinformatics. So can you see my slides, right? Before we start, I have 3 questions for you. You can answer through the chat or just think about it. We'll come back to this later. So the first one, how many of you have been working with neglected diseases? So doesn't matter if it's bench or computational research. Okay. The second and the most important, why are you working on this topic? Okay, think about it, hold that thought, and we'll discuss this later. And the last one, how many of you know or have ever seen someone with the disease you are studying? Okay, this is important. Okay, and we talk about motivation. So let's get started talking about computational biology and bioinformatics, or computational biology or bioinformatics, computational biology like / bioinformatics. I think sometimes these terms are used like interchangeably, and sometimes we can see a tendency of people saying bioinformatics, thinking more about next generation sequencing, or things more applied to translational medicine, personalized medicine, mutations, more genetics. And I feel when people say computational biology, most of the time they're thinking about more analytical methods and simulation and protein structure, systems biology. We have a lot of different terms here. I don't know if you have the same impression as me. I really would like to hear about this in the discussion section. But whatever the term you use, they have some things in common. The first one is it's really interdisciplinary field, and they both develop and apply computational methods in biology. And when we talk about this, usually are talking about large data sets or high performance computers. And they both are related to analytical methods in mathematical models and simulations at some level. Within these topics, I believe the one that most impresses me over the years is the interdisciplinary field. It's really exciting characteristics of the research in computational biology or bioinformatics. Sometimes I use both terms. I don't know how the platform works, but if you have any important questions during the presentation and you want to interrupt me, it's okay. Okay, I can talk about. Okay, so I like this one. As you heard, I came from physics. I have a degree in biological physics, actually. So I use it to work with more mathematical modeling in biology. So when you come from a more mathematical field, we use it to see more well-behaved things like this. I felt when I started to get more into biology and medicine, something more or less like this. Okay. It's really complex, and we have a lot of exceptions, and sometimes it's difficult to capture all the biases you can put when you're doing computational biology or bioinformatics. Some people try to convince me that the opposite road is more difficult, but I really don't believe so. They complement each other and need each other. Okay. It has been some years that people try to define the core competencies for someone to work with bioinformatics and computational biology. There is this interesting paper from 2014 when they try to create a summary of the main skill sets of a bioinformatician, hearing people posted jobs for core facilities or some research opportunities they saw on the internet. So we have a lot of different sets of skills. We can think about general skills, like the ability to communicate and to collaborate. We can think about computational skills in programming or work with distributed and high-performance computer. We need to know more about your specific field of biology that we're working on, whatever it's medicine or not. You need to have some skills in statistics and math. We can see R there, but I believe Python is also taking space now. And you have some topics, some skills that are really specific for bioinformatics, like retrieving and manipulating data from public repositories or analyze large data sets with really specific characteristics. If you work with genomics or if you work with transcriptomics, you have different types of files, different type of algorithms, and it's hard to keep yourself up with all the develop. It's really dynamic when you work with bioinformatics. Things change all the time. They develop really fast. So seeing this table that has some years now, you probably remind yourself about some recent job posts. Basically, this is a unicorn, at least in my view. You hardly find someone who fits well in all of these categories. So an interesting topic they bring in this paper, it's about the different profiles. So they use a simplified view in 3 different profiles. The first one is the user. So the user, you'd be someone who likes to distribute their work, like in bench, in computational work, and maybe have a preference for graphical interfaces and know really well the biology of what they're studying. We may also have like the science profile, the bioinformatics scientist in this case, who likes more to work in command-line graphical interfaces, and it's full-time dedicated to computational biology. And maybe you can think about a third that's an engineer or maybe a developer, if you're thinking about more recent terms. That's someone that's more dedicated to computational work, but more to like developing interfaces, developing algorithms, developing pipelines. It's more like the IT guy or the programming guy. So it's really an oversimplified view of a profile of a bioinformatician or someone who works with computational biology, but it's touch rise awareness of who are you and which is your profile. Of course, it's more complex, and you can walk around the different profiles we have here. But at least the first time I saw this paper, I remember when I started to work with computational biology, and I felt like I had to know everything. So it took some time and experience that to understand that's not really necessary. Sometimes you need to communicate well between the fields, and you need to complement each other. When you talk about bioinformatics, we are used to see some charts like this, but I have a bit different view that works more or less like this. So thanks for your render. We try to balance between the different fields and different sectors. And when we think about that, sometimes I will have a lot of trouble in communicating. I believe most of you had the same problem. Sometimes it's difficult to explain to the corporate IT that you need to transfer like 20 terabytes of text data. And it's difficult to explain to a physician the parameters and the quality control of a new diagnostic routine. So I believe we have to balance those things, to balance the areas, and have a really good communication between the fields and the sectors. That's one of the most important roles in computational biology and bioinformatics. So we don't need to know anything, but we really need to know how to walk between those different areas and those different sections, and bring new insights when you try to connect all those things. So when people ask about the corporate competencies, what you need to be a computational biologist or bioinformatician, I used to answer that first you need to understand your skills and the kind of professional you would like to be. You also need to understand the bioinformatic fields you would like to work on. And just then you can define the focus and the tools you need to learn. Okay, I'll give some examples. Most people in bioinformatics, if you talk about academic research, tend to use R. If you go more to industry and development, maybe people would tend to use Python. We have a lot of tools in workflow management, like Nextflow, the SnakeMake. So it's hard to just list a lot of tools or competencies that are needed if you don't know which field of bioinformatics you'll be working on. So you just get to know what you need to understand, what you need to learn, what you need to understand to start working with computational biology or bioinformatics when you start to talk with other researchers and graduate students, especially the graduate students that are also with you on this path. So you can talk to those people the fields are interested about, and get to know what tools they're using, when you need to learn R to work with you? Do I need to learn Python to work with you? What management workflow tools do you use? And you can also have that information in industry. It's interesting, this term, informational interviews. I don't know if have ever seen about this term. Informational interview is when you're interested in a company, like DASA, like I'm still a consultant there, or Novartis, or another one that you see that has interesting positions there. You can just maybe stalk a little bit the GitHub, the LinkedIn profiles, try to find someone that works there and ask to talk a little bit about their work. You may really get impressed of the amount of the positive answers you can get. People actually like to talk about their work. So with these informational interviews, we can get information on how's the routine of work in that place, which tools they use, and get a view if that's really what you want for you. Okay. So I did a lot of informational interviews, and some people sometimes ask me for ones, or if other bioinformaticians at DASA as well. It's a really interesting way to get in contact with industry if you're trying to transition for academic research to industry. We actually have a broad range of activities in computational biology. We can go through academic research, where you can see comprehensive genomic, transcriptomics, proteomics. We can start searching for biomarkers and signatures. We have protein and drug designing. And when we start to go more to translational research, that I'll talk a bit more later, or industry, we have a bit different range of activities. We can work on surveillance. Now we can may start to validate that molecular signatures or biomarkers that you found at academic research and put it in a routine. So we have validation, innovation, pipeline development. Some people work with user-friendly tools. There is a broad range of activities there. So I keep saying that's important to know where you're getting to. We also have another range of activities here, the third one, that's more server data administration, more computational work. So manipulation of data, the life cycle of data in bioinformatics, that's an important topic. If you're working with genomics, we have real large data sets. I don't know if we have an idea of the extent of the size of the files, but one run of NovaSeq data may produce like 2 terabytes of raw data. So it's not that easy to manipulate and administrate and transfer the data. Get a bit more into the topic of neglected diseases. There's a lot of reasons why people would like to use computational biology. I think the first and interesting one is we have it a cost-effective research. So neglected diseases usually affect populations in low-income regions. So that might lead you to think that we have less financial incentive for pharmaceutical companies to invest in research. And it's actually easy. We don't need a lot of to start with. And most people work computational biology at the beginning until they set up a lab and start to do more bench work. It's interesting to understand disease mechanisms. We can analyze large data sets with bioinformatics and computational biology tools. You can work in drug discovery and development, also in a cost-effective way. And interesting things, epidemiological studies. that you can use for modeling disease spread and predicting outbreaks. We have an example of the recent pandemics. A field that I'm working on nowadays, it's personalized medicine. This is especially important for neglected diseases because they often occur in diverse genetic populations like ours, so that involves human genomics too. And we have always possibility to work in collaborative and open sourcing efforts. I was always impressed that in neglected diseases, people are more open and collaborative than in other fields. So I will start to talk about two topics I have been working more related to neglected diseases a few years ago. That's in drug discovery and also epidemiological studies. So I don't know if I have a diverse background here, but we have different omics, that's informal term related to collective quantification categorization. That's generally associated with biological molecules. So molecules. We have genomics, epigenomics, transcriptomics, proteomics, metabolomics, and some of those omics may be related to real large data sets and the famous big data. So in my doctorate, I get into the field of structural biology. So I worked a lot with molecular modeling, molecular dynamics, classical molecular dynamics, and quantum mechanics. It's a field I really have interest in. So I saw you had a course on classical molecular dynamics yesterday. So you probably discussed about the 3D modeling. So which could be the simplest way to depict and to model a polypeptide chain or role protein, DNA sequencing, or even a membrane. So we have different types of models with different levels of details. And that depends a lot of the research questions you have that needs to be balanced with the computational cost of the model. So if you need to see some details of a protein-ligand interaction, you probably will need all atoms in your model. So you need to see your hydrogens, your hydrogen bonds, and all the details possible to work with drug design. But if you work with membrane or huge complexes, you probably will need a more simplified model to balance your computational cost. So in classical molecular dynamics, we have computer simulations to predict the trajectory, and you are using numerical integration of really the Newton's equation. I saw some pictures on your Instagram. You probably got into more detail about this yesterday and today in the morning. So it's a really exciting field. When I started to draw and see movements of proteins and fold complexes. That's really important when we talk about drug development, because this movement can really affect and impact the accessible area of ligands. So most of the time, people will try to combine classical molecular dynamics with virtual screening to work with drug development or rational drug development. We have different contexts here, like we can have, I'm not sure I can point here, we can have a protein and a set of ligands that well known, and you can go for a pipeline and select the most potential ligands for you. You can have your protein structure and have no idea what are the most interesting ligands for that. So you can screen like a million compounds. And it's really cost effective and reduces a lot the bench work. for experimental validation. So in the second line, we have like the structure-based virtual screening. In the third line, we have some null ligands, and they may want to find some similar ligands that may be more interesting or more effective. And if we don't know the target, we can find some potential candidates to that disease or that protein we're studying. So in my first postdoctorate, I worked a little bit in virtual screening and molecular dynamics with Paracoccidioides brasiliensis. So we combine molecular dynamics and virtual screening for the isocitrate liase. And it was really interesting to see the presence of cofactors and other ligands changing the solvent accessible area. And that gave us a wider scope range of ligand candidates. It took some time, but we could see that the closed conformation was leading us to ligands with more carboxamide and lactone moieties, and the upper conformation seems to induce more the presence of beta-carbonyl moiety. So we selected from the virtual screening 10 compounds. For two could not run these experiments, and most of them really inhibited the growth of Paracoccidioides, but especially one, both for Lutzi and Brasiliensis, we had a high selectivity index and an antifungal effect. So it was hard to, when I was working with that, and I started to realize that it took a lot of time to see practical applications of molecular dynamics and virtual screening. And when I was already working in another place, they called me and said, "Oh, it's working. The compound is selected. It's working." It's really exciting when start to see your computational work really get into practice. So this one of them, we also studied a little bit the null structure protein 1 of dengue and Zika viruses, also in the molecular dynamics context. So we got a lot of insights of important regions and flexibility for following this research start to work on the rational drug design. So we still have a consortium there, and people are still using these molecular dynamic results. As I said, it takes some time, like 10 to 15 years for a potential compound really pass all the steps that are needed to become a commercial medicine. But sometimes it's really to get motivated to work on that. But when we start to see that things are developing, it's really exciting. I stopped working with structural biology some years ago, and now I'm starting again a research group in computational biology and virtual screening, but now in the context of cancer. After my first postdoc, that was at UFG in Goiás, I spent some time at Barretos Cancer Hospital, so I started to work more in the cancer context, routine diagnostics and translational research. And after that, I spent some time at DASA, that's a medical company in São Paulo. Specifically on research, they had production and another ancestrality test. They have at least two different groups of bioinformaticians and also the research. And the first project I got was the genomic surveillance of SARS-CoV-2. I believe most of you get at least a bit in touch of SARS-CoV-2 research at some point during the pandemics. It was a really exciting project. The head was Dr. José Eduardo Levi, and they target like 30,000 genomes of the virus. Which is at least in March, we had like 22,000 genomes. That was like 10% of the Brazilian sequences. So it's a lot of work to do, a lot of pipeline development that now can be translated to another areas of research. So it was really interesting to see the global efforts on that, and that is something it's really would be great to continue it for other diseases as well. So the pandemic's impact on neglected disease, although it had a lot of negative impact, we have a lot of positive as well, because we could see unprecedented levels of international cooperation in health. We could see a lot of advancements in computational biology to try to see disease patterns, predicting outbreaks, and identifying new treatment targets. We leverage computational biology tools for drug discovery that definitely can mirror in the context of neglected diseases. We have had a lot of innovation in the healthcare delivery, like the accelerated adoption of telemedicine and digital health. And you can see a lot of this at our cancer hospital nowadays as well. And really interesting impact on vaccine development and distribution. People started to think about that, and we hope to keep that fostering the development also in translational medicine. So I cited translational medicine a few times. That's one area of research when you try you try to start people to work together in the academic field and in the clinical field and also in the community. So you try to get the research you're doing right now and try to think about how you could use that in practice. It's really viable to work with that, because sometimes you have a really good idea, but when we start to talk in multidisciplinary teams, they start to ask some important questions. Here I have some examples. You can have a question of the corporate IT, like how can you transfer and storage this amount of data? We're going to put like 200 terabytes. Sometimes the idea is great, but the nurse tells you that they're not able to collect this kind of sample. And the physician says, oh, this is great, but that will not really impact the clinical practices because this and that. And the technician says, oh, it's great, but the cost of production are really unreliable. And sometimes it's awesome, but too expensive for the health public system. Sometimes it's frustrating, but it's definitely better to understand these limitations up front. And when you have an environment, when you are able to have like this multi-professional meetings, you start to see these limitations right away. So it takes a bit of courage to expose your work and talk to people and communicate well about different things that are cited early in the talk. But it's really gratifying. So it's really great and exciting when you start to see things working. So the target of translational medicine is the personalized medicine. So it's a form of medicine that uses information about a person's own genes or proteins to prevent diagnosis or treat disease. So when you're not using personalized medicine, you tend to treat the disease with the same therapy. But some people may have a really good effect, take that medicine. Others may have no effect, and others even have a really serious adverse side effects. But when we start to understand that people are different and they may have different profiles and different needs, you can start to screening those people using genomics and try to identify those groups and also to search for specific therapies for each one. So, running away a bit about the topic, there are some types of cancer, like lung cancer, that already have these target therapies. So some people, that take just the targeted medicine, they respond really well, and some people don't. So you can go to more standard ways of care, like radiotherapy or chemotherapy. So you really need to know specific mutations and alteration that the person has to start the treatment. And you can reach personalized medicine search for molecular biomarkers or molecular signatures that can be used in the routine. So one example that we have here is in medulloblastoma, when the child comes for surgery resection. We can take a tissue tumor, we can do some experiment with a targeted set of 22 genes, an equipment called NanoString. We have an algorithm for molecular classification, and depending on the subgroup this child has, she can have less aggressive or more aggressive tumors, less aggressive treatment. So it's like if it's an WNG subgroup, the doctors know that that child will develop really well, regardless of the treatment. So she can have a less aggressive radiotherapy or chemotherapy and have more quality of life. So when we're talking about translational medicine and personalized medicine, we need to think about the role process. So this research is great, but it's really reliable to put that in practice. So this is extremely important also in neglected diseases, when you have a lot of difficulties to use what you're studying in the public health system. So I stated earlier that genomic diversity is an important feature. So neglected diseases usually are in more diverse populations, and I would like to highlight here that understand the human genomics is also important for whatever we're developing. Recently, I'm collaborating with the Genome Project. It's a populational genomics project from DASA. When you try to understand a bit more the Brazilian profile and to build a reference panel and working on polygenic risk scores. So that question I asked you before, why to study neglected diseases? You may think about want to make a difference in public health impact. You may be thinking about your professional development. I believe neglected diseases can offer unique professional development opportunities and also different challenges. And you may really be interested on the research, or you may be thinking about advocate for others and improve policies, reduce rough expenses. You can think about social justice and a lot of we can list a lot of topics here, but I believe it's really important for you to understand why you're doing that, because motivation really drives research. When you have a strong purpose, a strong motivation to work on your research project or your job position, I believe it doesn't matter. We can do a better job in the better research. I remember when I started at Bahis Cancer Hospital, there is one director that said to a few students that are not feeling like that motivated, that they should spend some time collect clinical data at the children's hospital, so they would understand better what they were studying and feel motivated when you get to really know what you're studying and what the reason, what's the motivate, what drives people to study that. So my motivation changes a little bit. Now I'm a researcher at Hits Cancer Hospital. You may have maybe heard about it. It was based in Barretos, but now have several units around the country. So we have 4 hospitals and more 2 on the way. We have a lot of mobile units to reach several difficult places in the country. And we also have the Molecular Oncology Research Center. So it focused on translational research. And nowadays it has more than 200 members and divided in more or less 15 research groups. We have a biobank of more than 250,000 samples. Good structure for preclinical studies in cancer cell lines and animal models, and also a laboratory of molecular diagnostics. I really welcome you to know a bit more about your institution of research. So thank you very much for hearing me, and I'm not sure if you have time for a short video. So
some say you'll never be alone. Some say that things cannot go wrong. But when you come undone, it's true. Sometimes you feel it's only in the dark. You feel alone. In the dark, all hope is gone. Dreams go by and fade away. Feels so cold, left us strange. Who can the forces steal? No dreams when you are in too deep. Sometimes you feel it's only don't give up, don't fall apart. Let the warmth inside your heart make you come back up again. Let the sun lead your way. As long as love fills your heart. You know you'll never fall apart. You know you'll never fall apart. As long as love fills your heart.
Thank you very much.
Congratulations for the amazing presentation. The video was incredible. Everyone is, I think, so emotional right now. So, once again, thank you very much. And now let's open for the questions. So, if anyone has some questions in the chat, just put it right there, and we'll project here for the Professor Lucien to answer. I have a personal question first. Uh, how challenging is it to work with virtual screening in neglected disease due to the lack of financial support in the research to obtain PTB structures and comprehending molecular mechanisms? Do you think the machine learning is the life-changing for this scenario?
Well, yes, we have a lot of challenges to work on virtual screening, especially when we don't know for sure or don't have a experimental 3D structure of good homology modeling for the target you're mirroring, you're targeting on. And I believe we have also in the another end when you actually found a lot of good candidates and you can go further with clinical trials and experiments and the only
It
thing we
gets less
have is just yeah, it's really expensive and takes years and sometimes they just don't have interesting that specific disease. So I
right.
think this is the two biggest challenges. I believe it's both ends. When you're starting and you really lack resources, and when you finally get it done, we can go further. So it's uh, it's up to you. I love the topic of neglected disease in computational biology because it may raise a lot of awareness about the topic and to maybe like give motivation to people like the one I have now to advocate for that, for better conditions, better funding, and maybe just get that fostering of the pandemics to get funding and investments.
Thank you for the answer.
Mhm.
We have a big question from Bruna. We'll split in two parts. So she said, great presentation. Could you elaborate on the challenge and considerations associated with the integration of diverse molecular data sets into vector screening methodology, and particularly in meeting the regulatory standards by different agencies like FDA, EMA, and Anvisa?
Um, I think that I don't have an example specifically in neglected diseases, um, but maybe I can give you one in cancer diagnostics that it's one more close to me, and I believe will like be in the same place here. Most of the studies are based in European populations. So when you start develop polygenic risk scores and diagnostic tools, usually you tend to find results European population. It may not work well for more diverse populations for like ours or for or African Americans. And it's um, it's hard to find the word in English today. um, that's approval because you can't know for sure if you don't have enough data for that population to start selling di- that exam, that diagnostic or that targeted therapy. So the time for us to get target therapies or better diagnostics, it's uh, kind of longer. So yeah, it's indeed a problem. I'm not sure I answered your question. I don't have a lot of experience on that, but I can see a lot of discussions over the years. It's a complicated point indeed. It's a really great question.
Thank you. We have a lot of comments, especially about the video. Everyone gets emotional, but we also have another question. Bibiana asks, after what we have been through with chloroquine and ivermectin, do you think there is a barrier with the medical class for the computer-aided drug discovery? I think more in terms of negationism uh, from the medics when they're talking to the patients. They're talking about medicine that was discovered from computational analysis.
Well, I think that depends a lot of the context. We are in a delicate period of time, so everything that the researchers said was like a bomb on the on the news. I remember when I was at the Genov project, and we found that some new variants or something different in the sequences. We had to report that to the state or the ministry. And we were really afraid to tell something because we couldn't be sure that it would be important variant, but we needed to report. And it was a really delicate period. But I don't believe that to be a barrier. I think things are more like uh, calm now. So we can start to see those things we find with uh, more time to validate things before they get into the news. So I don't think it to be a barrier or keep working with drug pre-proposing because that was the case, right? So we have something approved and start to screening what we have because it would be really faster to get into practice. But I think was special times, and I don't believe, at least I can't see that being a barrier for research.
Thank you very much. I think it was the last of our questions. So I want to thank you once again for the acceptance of our invite and for the amazing presentation to us today. So thank you very much, Professor Luciano. And that was the end of our first day. And as we bring the curtains down, we want to express our sincere appreciation for each one of you for the time and for the contributions to our symposium. And we are thrilled to announce that we will be back tomorrow at 3:30 Brasilia time zone for another day with a bunch of presentations and amazing talks. So set your alarms, mark your calendars, and join us again tomorrow for we continue to explore the bioinformatics solutions for neglected disease. So thank you once again for your participation. Rest well, and we look forward to welcome you back tomorrow for another day of our symposium. So good evening, everyone, and see you tomorrow.